So I started using drug-coated balloons about 2009. I got particularly interested in them in around 2012 and 13, so over 10 years ago, when I was lucky enough to be taken to Germany to visit the labs of Bruno Scheller, Klaus Bonaventura and Franz Kleber. And they opened my eyes to a whole new world of angioplasty without putting stents in. I then came home and decided I would very gently start working out how to do that in a safe manner in my own hospital. Within 6 months, my practice had changed, so I was DCBing at a rate of 50%, and over the next few years that went up to over 90%. The rest of my department then gently shifted, and since about 2015, as a group, we've been DCBing and stenting at around 50-50, which then gave us a great platform to look at the work that we were doing and follow some of the data that was already out there. So we are very heavily regulated in the UK with NICOR and BSIS, and they look at every single operator and every centre's outcomes. So for the first 3 years where we were using DCBs at stents at around 50-50, we got in one dollop all the 30-day risk-adjusted mortality for our centre, and we are judged against the rest of the country, and we had an expected survival rate, and our survival rate was 2 standard deviations better than that. So we felt that possibly one of the signals in there might be the fact that we weren't putting stents in so many people, which then drove us to start interrogating our database, which has thousands of patients in it, in more detail. We've looked at, for example, day case angioplasty, because people worry that you have to keep the patient in overnight. We did a consecutive series of patients, it's safe to send them home. We looked at dual antiplatelet therapy because people worry about how short the dual antiplatelet therapy should be with elective DCB cases, and it's still asked now. So a month is probably more than enough, and in the future may well be even shortened further. We then started looking at mortality rates, and that was particularly relevant a few years ago when there was a big issue around paclitaxel and very late deaths. We looked at all of our patients. We found a very clean cohort, elective patients, no previous angioplasty, no previous heart attack, propensity matched 2 groups, DCB only, stent only, and showed at long-term follow-up that the DCB patients were surviving significantly more than the stent group, which made us feel that we were justified in our approach. Subsequently, we've looked at left main stem only angioplasty, elective day case angioplasty, STEMI angioplasty, which is a little bit controversial, but was published last year in JACC Intervention, and was showing either equivalence or, in certain situations, a benefit to not putting a stent in again. And that's then taken us to the point where we're now involved in some of the big RCTs that are either running or about to start. So I think you've already removed the first hurdle. The first hurdle is people wanting to have the mindset to want to consider DCB instead of stenting everybody. Once you've got past that one, it's much easier. The first thing I would recommend, which is certainly what I did, was go and find people who are already doing it. Go to the meetings. There's now lots of information in the meetings about how to DCB. Once you've done that, I think the next step then really is starting in a very controlled manner in your own centre. When I first started, I was doing very discreet, very simple lesions, and I wasn't accepting anything other than angiographic perfection. And then you tend to learn really in stages. So most people would start in in-stent restenosis and small vessel. Once they feel comfortable with that, they start moving into larger vessels and bifurcations. They then realise that they're seeing a lot of dissections. When we start looking at the angiogram in a lot more detail, we learn that there is a lot in that angiogram that we're not paying attention to, but that can tell us what's safe and what isn't safe and what to do about those dissections. And when you've got people at that stage with maybe a slightly different, gentler approach to their angioplasty, the use of more specialist cutting and scoring balloons, you've got somebody that's already converted. So currently there's already a fair amount of data. What everybody's waiting for are the big all-comers studies involving larger vessels, because larger vessels haven't been attempted. I think because people felt that the larger vessels were a step too far initially for the wider community. Now I think it's at the point where the wider community want to see the studies for these big vessels. So there are already 2 big studies running. There will be a third one being set up soon. And by the time we're finished in the next year or two, there'll be perhaps 4 or 5 major landmark trials that will hopefully change guidelines. And I think that's what people are waiting for. After that, you then have to go into the more specific subgroups, and the areas of particular interest, I think, would be bifurcation angioplasty, because it's a lot more difficult with stents, very simple with a balloon. STEMIs, because they're a high event rate group patient, they're much sicker, and people worry about how easily you can restore the flow. And then CTO angioplasty, because there's lots of reasons why putting a small stent in is probably a really bad idea. So all of our data is with the Braun paclitaxel balloon. So that's still our workhorse balloon. We are involved in trials, so in trials we will use those other balloons that are waiting to see if they can prove non-inferiority or even superiority. So my general advice would be either use those balloons that are out there that do have good evidence. Some of them don't, you shouldn't be using those. And then the answer otherwise is to say to the companies, very happy to help win the study.